First, DNA gyrase is used to relieve the tension between the two parent strands. Next, DNA helicase breaks the hydrogen bonds between these two parent strands, thus separating them. And single-stranded binding proteins are used to keep these strands from winding back together. Next, primase is used to produce an RNA primer. These primers are needed because DNA must be built off of a free 3' end. DNA polymerase III begins at the free 3' end of the primer and begins to build toward the replication fork.
On the lagging strand, primase sets down another sequence of RNA primers. Since the lagging strand is more complex, it needs multiple sets of RNA primers because polymerase III builds from the 5 to 3 direction. On the lagging strand, it actually looks like it builds in the opposite direction that helicase unzips. Each fragment here is called Okazaki fragments. DNA polymerase I comes in and removes the primers and replaces them with DNA. This occurs once on the leading strand, since DNA polymerase follows the helicase on the leading strand, of course, since the leading strand is awesome and it goes from the 5 to 3 direction. But this will happen multiple times on the lagging strand. Once the primers are replaced, our fragments are not binded together. DNA ligase solves our problem by forming a phosphodiester bond between the two fragments and the other fragments. This comes to the end of our DNA replication presentation, and this is called semi-conservative DNA replication.
Our DNA goes through many coilings to pack into the nucleus of all our cells. When DNA is being packaged into chromosomes, it starts by the help of histones, which are protein subunits. 8 of these protein subunits cluster together before helping the DNA coil. Once the 8 histones come together, they attach themselves to parts of the DNA double helix. Once the histone protein subunits attach to the various parts of the DNA begins to wrap around the histones, creating a nucleosome. Many of these nucleosomes are created throughout the strand of the DNA. These nucleosomes stack on top of each other. This tightly packing is called a chromatin, which has a width about 30 nanometers. The packed fiber is further tightly packed by winding and coiling on top of each other, resulting in the creation of chromatin loops. The coiled chromatin loops supercoil by yet again folding on top of each other, creating condensed chromatin loops in order to allow the long pieces of DNA to fit in small nucleuses of cells. The end result of the tightly packed DNA is the structures that we can see through a microscope called chromosomes, which have a width about 1,400 nanometers. DNA goes into chromosomes only when dividing. Other than that, it is less tightly packed and less organized.